Okay, I'd like to get started. Welcome to the working group on multiscale modeling of immune function and the Global Alliance for Immune Prediction and Intervention, GLIMPRINT mini-seminar series. It's our May 14th meeting, 2026. And as always, I need to remind people that this meeting is live streamed and recorded and available on YouTube in edited form afterwards. Our list of people has changed today. We have Tom Helikar and myself as co-leads, Lorenzo Vischini and Ellen Quardakos as activities coordinators. We're always available to take your suggestions and comments. We'd appreciate your help in making this community a vital and successful one. Let us know what we can do better. I want to thank Jim Sluka from Indiana University, who's stepping down after 5 years of exceptional service as coordinator for the working group in GLIMPRINT. He's been web manager, he's been GLIMPRINT treasurer, he's done an amazing amount, and we wouldn't be here without his time, skill, and leadership. And we want to thank him for all the volunteer work he's done over the years and acknowledge his contribution. We have a variety of communication channels. They work better if you use them. We have a glimprint.org Slack channel, which doesn't get much use. There's an X handle, MSM Viral. There's the IMAG MSM Wiki page. There's the LinkedIn page, and there's the YouTube archive. Please help us develop and distribute these. We want to congratulate Yi Zhang and TJ Sigo, who are core members of the group, who were among the winners of the NIH replication prizes this year that were announced recently. Yi Zhang at Georgia State, winning idea to propose to independently replicate across multiple sites whether ATR FTIR spectroscopy combined with low complexity machine learning can detect MRSA versus MSSA within 30 minutes of beta-lactam exposure with 90% balanced accuracy. And then TJ Sigo won with an idea for assessing the reproducibility, replicability of published epidemiological models, including stochastic components. Congratulations to both of them. That replication prize was a great opportunity, and I'm so glad to see members of this community winning that. If you have a publication to share, those can be put up on the IMAG wiki site, and they can also be put up on the GLIMPRINT site. We want to thank Jim Sluka and Jacob Barak for all the work they've done on the GLIMPRINT site. Please contact Jacob or Tom Helikar if you have issues with the GLIMPRINT site. NIH has just announced a new area of interest in multiscale modeling, computational modeling of complex processes across biological scales. This is very much in our wheelhouse. The lead scientific contact is Reid Shabman, who has been a great supporter of this community and who has spoken to us on numerous times. I would encourage people to take a look at this area of interest and see what we can do to move this field forward. We still have the opportunity for people to apply to the 2026 virtual CompuCell workshop. If people want to do multi-cell modeling using CompuCell, we'd love to have you join us virtually. That's going to be between July 27th and August 9th this year, and we would be glad to have you there. We have 2 seminars in the books coming up. May 28th, we have Carolina Gonzalez-Carvazos from Scripps talking about drug repositioning with interpretable graph neural networks. And then Mario Acera from Barcelona talking about evaluation of single-cell multimodal data integration. You've heard me say many times that we would love to have your suggestions for people you'd like to hear from. You can suggest yourselves, your students, your postdocs. Don't be shy if you've spoken before. If you have new material to share with us, we'd be delighted to have that. Also, feel free to suggest experimental teams, computational teams, biotechnology or pharma teams, regulatory teams, anyone you feel could provide insights into the general area of immune modeling. They don't have to be modelers to speak here. They don't have to be doing immune work per se if it's ideas that can be translated. So please help us build this seminar series. You can send your ideas to me, jaeglazer@gmail.com. Lorenzo is very effective at getting people to come to speak. We want to keep this series lively and effective. Today, we're very pleased to have Kevin Flores from North Carolina State University, who is going to be talking to us about hybridizing data science and mechanistic modeling for biological systems. He's written many, many papers. A few of these are listed in the announcement here, and I will turn the screen share over to him for the seminar.
So yeah, thanks, James, for this invitation. It's nice to network with people that I don't usually talk to in the math bio community. So this is definitely a welcome platform. I appreciate it. Yeah, so I just wanted to give sort of an overview of some of the things that my lab does to hybridize what I would have traditionally called data science. Maybe it's called AI and ML now, with traditional mechanistic models for different biological systems. So here's just sort of like a high-level overview of the central problem that we face as mathematical modelers is that the data very rich, but they're heterogeneous and stochastic. Maybe there's spatial and maybe there are a bunch of late variables that we don't observe. And then we have these two ends of the spectrum where mechanistic models are interpretable, but there's infinitely many of them, or combinatorially large number of them. So it's difficult to parameterize that many and sort of choose between them. And I'll show a slide about that. And then data-driven models like AI and machine learning are flexible, but unfortunately, they sort of suffer from this interpretability issue. So the role of hybrid modeling, I think, is to really fuse these two to discover, select, and calibrate, and then perform uncertainty quantification for mechanistic models in sort of a semi-automated human-in-the-loop fashion. So you're going from biological data to hybrid inference to mechanistic insight. And I apologize for the emojis. My students are having me use more emojis these days, so I guess that's the way students communicate. So yeah, one of the areas where I sort of got interested in this was a postdoc of mine who started in my lab a while ago, John Ordini, had did its PhD about modeling reaction-diffusion equations for collective cell motion. And I think this really sort of epitomizes, the issue that many people have. So you basically have population-level models to encode that themselves and sort of encode biological assumptions about the way different cells interact with each other. Maybe there's some diffusion. Maybe they like to push away from each other. There's proliferation. So each term sort of on the right-hand side, we know sort of corresponds to a candidate biological mechanism that we sort of upscaled from an agent-based model in our minds to a continuum level. So the question is, how do we actually do this in practice? So in practice, this is the way that it's been done. This is the way that I was taught in grad school. So you start with some experimental data, you make a bunch of different assumptions. All those go into basically right-hand sides for models. With that, you have some fitting procedure. If you're a Bayesian, you use something like ABC these days. For your frequentists, you use some sort of generalized least squares framework, but in the end, that gives you some model selection scores, either AIC or BIC, or possibly nested selection scores, for which you get the best AIC score, which gives you sort of like the final model, and then that's the one that you're interested in. So this is sort of typically how it's always been done, and so we want to sort of speed this up, right? So three hybrid modeling interfaces that my lab has worked on are sort of equation learning, which in the broader sense is just how do you discover interpretable ODE and PDE structures directly from data, or more likely a candidate set of structures that you would then need to go fit with traditional like mechanistic modeling parameter estimation methods like ABC, for example. The second one is topological summaries in ABC. We found this particularly useful for summarizing particle data, so I'll talk about that in a little bit. And also another way to sort of add a little bit of flexibility in your ODEs and PDEs is to think of your parameters as parameter distributions, and sort of fit a distributional inverse problem. And so I'll talk about this Prohorov metric framework in this Randy code that's on GitHub. So yeah, so for the first one, equation learning, we have this paper called BINS, which is, I guess, 6 years ago by now. So biologically informed neural networks, they're basically an extension of physics-informed neural networks. And what they require you to do, and the problem that we were trying to solve here, is that when you traditionally applied equation learning methods, you would have to sort of come up with a library on the right-hand side of, maybe hundreds of terms or something that you'd want to select biophysically relevant mechanisms from. So instead of doing that, this sort of goes halfway between that and having to fully specify the structure. And so what it says is we can specify sort of like a flexible structure. We basically know the cells are diffusing somehow, and they're moving around and they're growing as well. And so we're just going to replace those with MLPs and jointly optimize the parameters for both the cell density over time and the diffusion and the growth rates. And the goal of this, again, is not to take the human out of the loop, but to put them in it to be able to look at these graphs for D and G, and then sort of guide a mechanistic interpretable structure to that. And I'll show that in a second. So just break down typically what the loss function structure for these types of problems is it's usually a multi-objective loss. So if you go back here, you sort of see this neural network architecture. So it's got three sort of pieces to it. The first one is just your usual fitting to the data. So if you have, for example, a weighted least squares error model, something like higher your observation is, the more noise you have, then you can sort of fit that in a generalized least squares framework. The second term is typically something like an ODE or PD constraint. So from the actual fitting of the neural network surface to the data, here you see UMLP, you can then perform automatic differentiation on that with respect to time and space to sort of form what you think should be the left-hand side equaling the right-hand side. So the reason it's minus and squared here is you want that to be 0. And so finally, one of the sort of unique things about this paper, I think we were one of the first to sort of think about, is that we can also enforce some biological assumptions here. So for example, if you assume like diffusion is always increasing with respect to cell density, or growth is always decreasing with respect to cell density, these are sort of reasonable assumptions that you could sort of put in there. And so the user can then, sort of construct this constrained loss function and sort of add that into the multi-objective loss. What we found is we applied this to some scratch assay data from Matt Simpson's group at Queensland University Technology. And he had been trying out various Fisher KPP equation formulations. Some of them were sort of nonlinear. But this sort of shows the problem here, where if you try to fit all these different models to this scratch assay data, so you basically make a scratch wound, and then you sort of watch a cell sort of, I think these are breast cancer cells, fill up the wound. You still get quite a lot of model discrepancy, especially at the early times in this experiment. So what we did was we applied this BINS framework to this. And then out of it, you get, first of all, a lot less model discrepancy. And then second of all, you get sort of these, there's the model discrepancy part. And then you get these interpretable graphs. So these graphs are then used by the mathematician to then go basically hypothesize what functions should be on the right-hand side. And I think this is probably the healthiest and most useful way to use these hybrid methodologies, not to, again, completely replace the mathematician and thinking about the mechanisms that are there, but then looking at these graphs and saying, okay, I think this is a reasonable set of assumptions. And so if you do that over all of the different replicates, these are the sort of graphs that you get. And then this one is an outlier for various reasons. But these are at different initial densities. And so this approach is working really well at these different initial densities. And what you're finding here is there's basically non-zero baseline diffusivity. Everything seems to be increasing and concave up. Everything for GMLP seems to be approximately linear, which makes sense. It's basically a logistic growth model. We also needed a little bit of a time delay in the beginning to allow the cells to sort of recover from being scratched. They don't like being touched. And so you can then go ahead and posit what kind of reaction diffusion model might actually fit those type of data, so that you can go ahead and have a baseline diffusivity with a nonlinear increasing concave up function for your diffusion, your typical logistic growth model, and then logistic regression function does reasonably well for this sort of sigmoidal time delay. And then when you go ahead and actually do traditional model comparison on sort of this bin-guided PDE, these are the results that you get. And what's pretty interesting about this in comparison to sort of like previous efforts on this data set is that there seemed to be the largest improvement for highest initial cell densities here with regards to the authentic information criteria. Okay, so recently we've been sort of extending these ideas, and this is a paper that was, I think, literally published last week, maybe, in PLOS Computational Biology to multi-experiment equation learning. So this schematic here basically summarizes the mean-equal ideal. So starting from applying equation learning to upscaling agent-based model data to differential equations, which is sort of the goal of this paper. On the left-hand side, you're sort of starting from this agent-based model, and on the right-hand side, you're hoping to have a generalizable equation-based model that sort of generalizes out of sample across all parameter regimes. So this is a pretty interesting study because basically what we found was in different parameter regimes, different models seem to fit better or worse. So you can read the nitty-gritty of the paper. There's like 10 figures in the paper and a bunch of supplementary material. But just to get the idea across, again, on the left, you start from agent-based model experiments. You run those at different initial conditions and different parameter values. And in the middle here, we show different ways of basically doing equation learning, trying to coarse grain those ABM simulations into an ODE model. So at the top is the classical approach. You can use the mean field equation. So it's interpretable, but it usually is too strong of an assumption, basically has almost no spatial structure. So anytime there's spatial effects, it's going to influence the dynamics. The next two boxes here are the mean-equal approach. So the first one is called the embedded structure approach. So we basically try to constrain the library on the right-hand side to learn one unified model across all parameter regimes at the same time. So the model has shared structure, embedded structure in it. And then there's also this one at a time approach where we basically apply equation learning one parameter set, one initial condition at a time. And then we do some sort of fancy interpolation across those learned terms across parameter space. So in the end, that's basically what you would get is a generalizable model across all different parameter regimes. And at the bottom here is the sort of traditional equation learning without generalizability. And so the right-hand side here sort of shows you exactly why this might matter. So parameterized ODE model can actually be tested on out-of-sample prediction. So in other words, if we change the parameter value, can the learned equation actually still predict, for example, population size, or for inverse problems, given new agent-based model data, can we actually infer the agent-based parameters that actually generated it? So I think one of the most interesting figures in our paper is figure 10 here. So here we're asking the question, can the learned equation model do more than forecast trajectories? Can it actually like recover the underlying ABM parameter that generated the simulation? So the parameter of interest here, it's only one. We only changed just the proliferation rate. And so this is on a birth depth migration model on a lattice. So the figure basically compares three models here, the mean field model, the embedded structure approach, and the one at a time approach. And then we only train these models on basically a sparse grid of 10 parameters between 0 and 5. And then we basically had it sort of like interpolate the learned equations across this wide range here. So that's what you're seeing here. The bars here show the relative error over repeated ABM simulations. So the main point is that, simple mean field models work reasonably well when your proliferation rates are relatively small. I think that's relatively well known in the literature that this is basically the mean field regime. But as proliferation becomes larger and spatial effects actually matter more, this equal approach and the equal approach do substantially better. So the whole point is, the learned model isn't just descriptive, it could actually become a practical surrogate for ABM parameter inference. So another area that we're getting into right now, this is work in preparation, hopefully come out next month. It's part of the NSF-sponsored IHBM program that I'm part of. So the central question here that we're asking is whether we can take a detailed infectious disease agent-based model. Here we use this published model that was validated called COVASIM, and try to learn a lower dimensional ODE model structure that preserves the most important sort of epidemic dynamics and intervention effects. So COVASIM was particularly useful for us here because it allows us to include things like inter-individual heterogeneity, contact networks, things like quarantine, testing, contact tracing, and so on and so forth. But those same complexities are what makes it difficult to actually directly analyze using tools like sensitivity analysis and optimal control and uncertainty quantification, which is why we want to try to learn an ODE structure that sort of preserve, make sure that it's as accurate as possible for those purposes. So what we're doing here is we're using our BIN, our biologically informed neural network, as a bridge between the ABM and an interpretable complemental model. So instead of assuming that key rates are constants, we basically let the biologically informed neural network learn state-dependent functions from COVASIM simulation data itself. So for example, I'll show you on the next slide, the effective transmission rate and contact tracing rate can actually depend on the number of susceptibles, asymptomatics, symptomatics, quarantined individuals, as well as things like the intervention strength. So I would just want to emphasize here that the reason that the BIN is actually useful is that it allows us to simultaneously impose a complemental ODE structure while still learning some of the structure of the ODE as unknown functional forms for the data. So the neural network is not supposed to replace the epidemiological model here. It's basically estimating the parts that are hard to derive analytically from the agent-based model simulations, with the long-term goal, of course, of producing fast interpretable surrogate models for whatever complex SDIR type agent-based models that you might have. So just a little bit more details on this. So here, the H and the B are the hidden observed infection and related latent dynamics. So that's approximated by a BIN. And the B as well, behavior and intervention related dynamics going from E to QE are also learned by a BIN. And then I think one thing that is going to be sort of interesting for people here is that what we found is that if you just apply the BIN and then apply sort of like a symbolic regression technique to learn symbolic structure of the OD on the right-hand side, what you end up getting is actually not you get sort of a first best guess of what the structure might look like and what the parameters might look like. So that's the sort of blue line, or sort of the blue shaded area here, which we're calling the prior 95% confidence interval. But when you put that through another procedure, like an approximate Bayesian computation, to actually do a refinement of those parameters after you fit those symbolic functions, it gives you actually like a much better estimation, prediction of what the out-of-sample data actually are. So the green here are the out-of-sample data for, it's just 3 compartments. There's like 9 or 12 compartments in the simulated data set here. And then the posterior mean trajectory here is in red with the 95% confidence interval in brown. So yeah, one of them that we've also been working on a lot is using topological data analysis as sort of summary, not summary statistics, but to summarize the topology and geometry of particle interaction data. So the main motivation that we had here was, we were collaborating with Jason Hawk, who's in chemical and biological engineering at NC State. And he usually runs his experiments. Okay, that wasn't a very long video, but you could sort of see all the cells sort of like exchanging places, intercalating with each other. And these are fibroblast cells growing at very high density. And what you could see is something that I thought was pretty amazing, which is that, cells aren't just bumping into each other randomly like billiard balls here. They're actually like actively using their shape with their neighbors to sort of like exchange locations. And they're doing so in streams and sort of like in parallel and anti-parallel matter. So if you do the single cell trajectory localization, you can sort of aggregate things over this video. and see that the relative angle of the neighboring cells is more or less parallel and anti-parallel. They're sort of streaming back and forth between each other. So our main question is, and I've been working on some topological data analysis to summarize angiogenesis models, whether we could apply the same techniques here to summarize the topology of these data, and then basically use that to fit agent-based models. So doing that with ABC. I'm not really a good agent-based model builder. So maybe that's something I could get from feedback for you guys, or maybe I could send one of my students to your guys' summer program later this summer. So we just fit this traditional Thorstonian model. And the reason that we did that is because we know that this model in the literature produces lots of different qualitatively different sort of global behaviors for just 2 parameters. So that was the main reason that we used it, not because it's mostly like biologically realistic for these data, but because we just between 2 parameters, we can get like lots of different things to happen. And so just to dive into exactly what is going on here with TDA, typically what you do is you have some sort of persistent homology summary. So here on the x-axis is time, and on the y-axis is what's called a proximity parameter, and it makes these Crocker diagrams. And so it's a time varying diagram because on the x-axis it's time. And it's topological because, as the title says here, it's summarizing Betty 0 and Betty 1. And you can also do higher order Bettys as well. Betty 0 is more or less the connected components, and then Betty 1 is the loops, the cycles that are in a graph-like structure that are sort of created when you overlap epsilon balls at increasingly larger radii. So that's what's going on with the proximity parameter. So I won't attempt to explain all of TDA, in like 1 minute, but more or less, what you get out of this is almost like a topological fingerprint or barcode for these simulations that's very robust with respect to noise. And maybe I'll just explain this one a little bit more. So for example, as you expand an epsilon ball around all of your data points in your point cloud, as that ball gets much, much larger, everything is eventually going to oversect and create like one large connected component. So that kind of makes sense why these figures look this way. But the interpretability of what's going on with Betty 1 and Betty 2 and so on and so forth is very interesting, and it's a problem that we're actively looking at right now. So just to convince you that persistent homology is a good characterization of all the emergent behaviors that could happen in an agent-based model, if you take those Betty 0, Betty 1 plots for every single combination of C and L parameters, you can think of it as like 2 matrices concatenated together, and then you pull it out into a long vector, essentially a 40,000 dimensional vector. You can compress that vector down into 3 dimensions by various methods like PCA or TSNE. And then you can treat those 3 dimensions as basically RGB dimensions and color this plot. So this plot should not be colored. It should just be gray. You can color it by treating them as RGB, and then you can map those RGB colors back onto the original CNL space here. And what you get is something that looks very close to the bifurcation diagram, global bifurcation diagram for this model. And I'm just sort of picking and choosing various points in CNL space and sort of showing you that the fingerprints look different, and then the sort of dynamics also look different as well. And I think for the sake of time, I'll just skip the approximate Bayesian computation part of it. But the upshot of it is you can then basically simulate these data to generate like synthetic Crocker diagrams. And then, of course, you can compute the actual Crocker diagrams from the real data, and then you can match them together. You can use various methods to do that, but we found approximate Bayesian computation a preferable technique, in particular because you don't need to calculate a derivative of things, and the Crocker matrix is not differentiable. So this is just a fancy way of just saying we're just trying to match the Crocker matrices from the simulations and the experimental data together. And so we also have a paper that's in review. This is actually on bioRxiv right now. We go a little bit one step further, and then also perform model selection. So not only can you fit one model, but you can fit many different types of models to it. And then afterwards, you can do your usual BIC model selection as well. So this is the, you want to look it up on bioRxiv, it's called Topaz. All right, so in the last few minutes here, I just wanted to bring this up because I think it's something that maybe not many people know about, and I just want to sort of spread the knowledge about this, that it even exists. Um, and that's that you can add a little bit of flexibility into a known structure of any ODE or PDE model in the form of what's typically inter-individual demographic heterogeneity. So for example, I always start with this Fisher-KPP equation because it's, just a really good sort of canonical example. You can take the D and the rho here, and then you can actually treat them as random variables. So if you assume like not every cell or individual in a population acts exactly the same, you can replace those with the distribution. And you might think, oh, do I have to replace it with like a Gaussian or a log normal? And the answer is no, you don't actually have to replace it with any distribution at all. You can just make it flexible, and you can actually learn what that probability distribution looks like in a non-parametric way, non-parametric because you don't need to specify the distribution automatically. So if you want to take a look at the details of how you do that for this particular equation, basically what you have to do is formulate an expectation over your probability distribution, and you have to sort of numerically discretize your distribution. There's a couple ways to do that. And yeah, you basically learn the weights of the numerical discretization. And then the output is the usual expectation of work, because this is actually what you end up observing. So you can add a little bit more flexibility into your models by assuming that not every individual acts exactly the same, which is honestly the most realistic assumption you could probably make about biological data, I would think. And we also have an extension to this to interphenotype competition models. You can take a look at this paper. I think you more or less get the idea. And then we tested it on a couple of synthetic data sets where we're learning go or grow subpopulations here. So there's some simulated parameter densities. So these are actual like densities that we're pulling parameters from to sort of simulate the entire distribution of individuals. And then, you can do your fitting. We found it's much more computationally cheap than trying to fit like a 2 or 4 or 6 subpopulation model, for example. You can just do it sort of automatically by just like learning the distribution, and it's fairly accurate. And then this is the recovered distribution. It doesn't look fantastic, but at least it can tell you that there's 2 different Gaussians going on here in D and rho space. And then we're just showing here that you try to do a similar thing with like a 2, 4, and 6 PDE subpopulation model. It sort of fails. And so we'll plot telling you how to do cluster selection. Okay, so that was a lot, but hopefully it's good motivation for some discussion. Hopefully I've sort of convinced you that hybrid modeling, I think, is the way to go. It can allow you to turn, your biological data into mechanistic hypotheses using things like equation learning, TDA, definitely applying some refinement after you're doing equation learning with ABC. And also, just so you know, there's this thing called distributional inverse problems for which there's a big history, and there's many books written about it. I never knew about it until I was a postdoc, so just letting people know about it. And I think really the goal of the way to move forward is to not think about replacing mechanistic models, but actually strengthening them by hybridizing them with these various methods from data science. And I'm always interested to know ways that people are doing that, and happy to have conversations about that. So final slide is acknowledgements. Just want to acknowledge my current and former lab members who contributed to various parts of this, and collaborators as well. I was always given the advice by my mentors that you should surround yourself by people that are smarter than you and better than you. And I've definitely achieved that, I think, with all these folks. And this is just a picture up here of a recent outreach event that I'm pretty proud of. It's my grad students explaining various concepts to K through 12 students at the North Carolina Science Olympiad. There was a couple thousand students that came to compete, and they were all very, very interested about biomath, and we need to spread the good word about biomath. Still, there's a lot of kids and parents that really don't know much about it. So as much as we can do to spread these concepts and kind of get people interested. You can see this kid here is like scratching his head thinking about this problem, sort of inspire us to communicate a little bit better. And of course, various funding organizations, especially the NSF, for allowing me to have the flexibility and the freedom to sort of pursue these ideas in various grants. So with that, I'll just take some questions. Thank you.
Okay, thank you very much. It's a lot of material. Their floor is open for questions. Yi, please go ahead.
Hi, Kevin. Brilliant talk. Great. I have a question about the last example, just 2 slides back. When you're talking about those Gaussians, PDEs, right? They represent non-interacting subpopulations, right? Does your method also work if the subpopulations interact with each other?
Yeah, that's right. So this is non-interacting, and then this is the way that we solve the interacting problem here by putting the data itself into the total population size. So the previous postdoc of mine, Erica Rutter, actually recently wrote another paper with the ODE formulation where they applied this to like real cancer data, cancer cell lines, and then they showed that, you know, it's able to accurately, or actually not accurately, but because you didn't actually know what was in there, but is able to recover like multiple subpopulations, and that those models were actually better than like single subpopulation models using this Prohorov metric framework. So definitely go and check out her papers.
There was a request that you please talk briefly through that Bayesian slide that you skipped.
Yeah, so what's going on here with approximate Bayesian computation is we're just trying to estimate a posterior all the time, right? So you've got notation here is Q for parameters. I'm using this upper script 0 for observed data, and then we've got our simulated data. So the problem here always is sort of calculating this intractable likelihood. And so the way that you do that with Bayes' rule is you can, well, we can flip things around, but then you basically insert here a pretty big assumption, which is that this likelihood can be replaced by this sort of threshold. So you're calculating a distance, and that distance can be calculated in various ways. Here we calculated just basically with sum of squares error and some preset tolerance. And there's a nitty-gritty about how you choose this tolerance as well. But basically what's going on is you're replacing the likelihood with this threshold distance in here. And the reason that we like this for this particular problem is that these are things like we can pop out of the agent-based simulation that come out naturally that we can compute. Whereas taking derivatives and things like that, although we are working on methods for doing that as well with neural networks right now, is much, much more difficult. And so, yeah, what happens is you basically just throw away from the simulations or you reject things that are, I think that should say feasible instead of infeasible, my fault. You throw away things that are having too big of a distance away from your data. In this case, it's the summary statistics, the Crocker matrices, and then you're keeping parameter values that are having close Crocker matrices with your actual Crocker matrix data. So yeah, it's not too complicated. It's just basically a forward solve, do a bunch of it, generate a distribution using this acceptance rejection metric here. And the way that you sort of construct this, people do it in different ways, but this works for this data.
Hi, thanks for the talk. So the question is like, when we are modeling a highly nonlinear, large number of variable ODE model, especially we want the model to be in steady state steady state with the physiological concentrations. So what would be the better way for parameter estimation, especially when we are including Hill functions in the ODEs?
I think, yeah, so I think the question is if your data are actually not very information rich because you more or less have only steady state information. So at t equals infinity, for example, how do you actually estimate things for the transient behavior, right? Like that these Hill functions actually activate at like the half max of things. And then the answer is probably it won't work, I think. But I think if you're looking for diagnostics for when it will work, you know, there's lots of books written about things like Fisher information matrices and calculating covariances and looking at their sort of sensitivity of those basically summaries of those matrices. Like you can look at the spectrum of eigenvalues
Right.
with respect to like sampling at specific times. So this is a whole like field of like optimal experimental design theory.
Jacob, please go ahead.
I have two questions. One of them is, at the beginning, I'm seeing that you're using both PDEs and neural networks and ABMs. It's like all of those, like too many, it's too complicated. And ABM has a power on its own that's like, you can basically replicate differential equations. Why do you need to do both of them? It's like, I'm trying to figure out like, why the added complexity? This is the first question.
Our motivation is always scalability. So this agent-based simulation that we chose here, for example, for CovaSim, is relatively quick. You can run a simulation in just a few minutes on your laptop. But we're really thinking about if you would scale it up to like a high performance computing that's required to do something like 300 million individuals or something. So if you're trying to do that, you know, there are methods to, I guess, coarse grain analytically any type of ODE model, but I don't know if you would want to go about it, you know, with this type of model here, right, where you're sort of unsure about which terms actually should be coarse grained and which ones shouldn't. So this is sort of an automated method, I think, for determining when spatial correlations are, I guess if it's network heterogeneity, we haven't really looked at that, are really important for doing that upscaling to the surrogate model that runs faster.
3 million today is nothing. It's really, you can do it on your laptop multiple times, really. So it's not a big deal anymore. No need high performance computing anymore for something. For 3 million, no. Anyway, but that's a different thing. It's like I didn't understand why the complexity, but now I understand. It's like, but by the way, CovaSim can handle it very quickly. It's a fast system. And my other question has to do with the neural networks. What kind of neural networks are you using, and what's their purpose? Like, which layers are you using, and what are you trying to optimize there or train?
Yeah, that's a great question. And this is a question I think get from reviewers a lot. So the question is, I think, two things of what you're asking. One is like how many layers, how many nodes, and which activation functions do we use if we're using vanilla neural networks?
Yeah, is it a feed-forward neural network, a CNN, LSTM? I'm trying to figure out what you're doing.
Yeah, so this is just plain vanilla neural networks. And you can do automatic selection of things like sparse regression. But actually, there are even fancier things that exist in this where people use Gaussian process models, neural ODEs, deep nets, and yeah, there's lots of fancier things that you can do. But I mean, the upshot of all this is it's just taking high dimensional data, a lot of it, and then trying to compress it into a low dimensional representation, which we're
Did you
forcing
try a
into the specific
transformer?
left and right hand side terms. I did not try a transformer. No.
Okay. Try to plug in the simplest transformer. Try to plug in BERT. See if it gets any better.
Okay.
Okay, Yi, please.
Hi, Kevin. It's me again. I have another question about your TDA. Your example is fibroblasts, where you see individual cells, mesenchymal cells. What if I have a tumor growth example, right? When you actually have big clusters, you have various sizes of clusters. And can you comment on, in those kind of situations, especially when you go through EMT, can you comment on TDA compared to other spatial statistical metrics like RePreS-K or 2-point correlation, all those things?
So I think if I understand your question correctly, and I think it's really interesting avenue of research is if I didn't have an agent-based model, but I had maybe like a continuum model, which itself has some spatial structure in it, because, you know, you could, you know, simulate whatever Turing instability or something like that. How would these methods apply to those types of data, like a like sort of like a larger scale, or even if you just had real experimental data where you're really not getting where each individual cell is, but you're getting like a dense density of clusters of cells or things like that.
Exactly.
So I haven't tried it at that level, but my general impression is like it would probably work. And actually visiting a lab in Kyoto University next month that does exactly that with spatial models of different skin diseases. So these are like couple multiscale models. Anyways, I don't want to speak for them, so because I don't do that research, but there are people who have sort of looked at those things. Also, you should look at papers by our collaborator Alexander Volkaning, who's
Yes.
also worked on ABM and
Yes.
PD models for zebrafish pattern formation. So those would probably be the closest things. But
So
I really like TDA because it has the
TDA,
ability
right?
to integrate. Yeah, still TDA. That's right.
Yeah.
Yeah, I was just going to say I really like it because it has this ability to sort of integrate local to global information in these Grok matrices. And one sort of interesting kind of user flexibility that you have is the ability to define different filtrations. And I think this is something that is probably an interesting avenue for research for people in math biology, where here I just showed you the usual Vittor's Rips complex, this idea of just making epsilon balls and then making simplicial complexes from it. But in another paper that we have with this angiogenesis model, we came up with a couple of different ways at looking at static simulated blood vessel network data. And we basically needed those other ways that we created these filtrations, basically looking at the data from left to right, right to left, top to bottom, bottom to top, and so on and so forth, in order to get really like accurate parameter classification and stratification of parameter space in those cases. So it's it's not obvious, I think, which filtration, so to speak, of ways of constructing these persistent diagrams works best for any given data set. And so I think that could be an interesting avenue of research. And if anybody has any thoughts on how to solve that problem, I'd be interested.
I was going to comment, since you have the TDA slide up, we often think about using machine learning for classification to avoid doing feature-based approaches. But in a sense, the topological data analysis, because it returns such a large space of information, is not as limiting as a lot of the classical feature selection algorithms.
Mm-hmm.
So what do you think about, in a sense, doing classification directly from images as opposed to going through the topological data analysis method? What are some of the pros and cons?
I think really it's just the interpretability of it. I mean, in reality, like it would be really nice to have just whatever CNN or tell you what the parameters of either an agent-based model or ODE are given some data. But I really do think like probably the stronger thing to do is to do some sort of in-between pipeline like this, where you build an ABM and then extract these features, extract similar things from your data, and sort of give it like this feature training because you know that these things are important. Whereas a CNN might go off the rails and be a little bit too unconstrained. Here we're sort of like focusing it on really just like the topology of the interactions between the different agents in the model. And yeah, one of the things that we're working on right now, which I'm really excited about, is basically doing the ABC within an entirely differentiable framework with neural networks as well. I think with that, we're going to be able to propagate back to the original agent-based model itself and really know like which parts of these Gawker matrices correspond to like which parameters and terms, if that makes any sense, kind of do a sensitivity analysis.
Well, I would love to sit down with you and take our angiogenesis simulations and run them through both the topological data analysis approaches and also the biology-informed neural networks approaches. We've been trying to do the kind of classification directly, and would be really interesting to see how using the TDA change the efficacy of the parameter inference in those situations?
Yeah, I would say if you could wait like maybe 1 or 2 more months, I think we'll have another paper out about doing exactly the sort of differentiable ABC framework that we talked about.
So there was a question in the chat which said that you have published really a lot of papers. Can you provide 1 or 2 papers that you think are the best ones to get started with these methods?
Yeah, I think the one that was in the announcement that you showed on the slide before I gave my presentation, the one that says the Royal Society interface paper, it's actually a review paper that was supposed to be written like a tutorial review, and I think a lot of people like that paper. It's basically about learning ODE models from agent-based models, because it gets the point across and sort of like shows the limitations and what you gain from doing this equation learning approach. So I would probably start there. And then second paper, I would read the Ben's paper, which, like I said, I think since 2020, there's a lot of fancier methods for doing the dimensionality reduction part of it and sort of filing it into these right-hand side terms. But I think that paper more or less gets the point across really well.
I'm excited to think about all the different places you could apply these methods.
Yeah, me too. I'm currently here at the multiscale immune systems modeling. We're having a retreat these 2 days, and there's just so many opportunities to apply this to spatial codex data. So I'm just happy to be even a part of this. Feel very lucky over here.
Itamar, sorry, did you have a question before we break?
I'm really overwhelmed and impressed by this barrage of methods, and all power to you, but I'll be very naive asking about the 2 Gaussians mixture. At some point, it was invoked as a very good mathematical trick. And I don't know if you're using it in that sense, but I'm asking a very naive, intuitive question, intuitive in my frame, which I'm not an expert. Can every Gaussian that you put in the mix, maybe more than 2, can be used as a source term, representing a source term in the equations, and then these two or more Gaussians interact, as was mentioned here in passing, and maybe it can simplify things because of that power of the trick.
Yeah, I think I see what you mean. We haven't really thought about it like that, but yeah, maybe if you think of it as inputs or something, there would be some simplifications coming from like control theory or something. Is that what you're thinking?
Yeah, if a Gaussian represents some source that is inducing some situations, like several scenarios, then another Gaussian counteracts it and so on, and it can be fun.
Yeah, I think we may get into some of that here with this whole multiscale modeling center. I'm just thinking about it because I'm here in this conference center right now, where one of the powerful things about this hybrid framework that I've discussed is it really gets you into the idea of using AI and data science to be able to use traditional approaches. So it allows you to use optimal experimental design theory or optimal control theory and things like that. So once you get from data to hybrid model to actually symbolic model, then I think it could be really interesting to look at these questions.
Beautiful.
But yeah, I think you're certainly right.
I have one more. Since you mentioned dimension reduction and classification, there is UMAP, of course, and Disney. And it could be that we can follow differential equations like time evolution and produce different data sets at every time step, and then do UMAP on those and see how the classification varies in time, namely certain colored regions will go away from one another or go close to one another. Am I wrong?
Yeah. So I don't know if it's related or not, and maybe you can check out the Topaz paper on Fire Archive, but what we have in that paper is another term in the model. So the Darsonia model has changed structurally. And so usually that term is 0, but then as you sort of increase that term even ever so slightly, like it actually changes that whole colored topological global bifurcation diagram. So I think that's maybe what you're talking about. There's some sensitivity with respect to parameters. Is that right? Or structural terms?
Yeah, maybe even this will help to understand this human mystery. The
Yeah,
success is quite mysterious.
I agree. Yeah, I think if you can get away with it, just use PCA. I agree.
Okay, thank you very much.
Thank you. I think everyone has had a good discussion. And Kevin, I really appreciate you coming. And I will look forward to hearing how things progress. And again, I will see people in 2 weeks. Thanks, everybody. Bye.
Thank you. Bye.